In the 21st century, we send humans to hang out in low Earth orbit. It's just a thing we do now. It's an incredible scientific and engineering achievement. No need to make a big deal out of it. But more recently, scientists have been sending ultra-mini versions of our organs to space, too. We've sent hearts on a chip to space. We've sent kidneys on a chip to space. We've sent mini brains to space, albeit super simplified ones. And next year, we'll be sending mini livers. And I wanted to know, what gives? I mean, don't get me wrong. We want to send a mini brain to space and see what happens. I mean, it doesn't need much justification. I say it out loud, it's immediately obvious. It's a cool experiment. Let's do it. But let's also talk about why we might want to send mini brains to space, what these mini organs are, how we're sending them up there, and what we could learn by doing all this. So why are we doing all this? Because the human body does not respond well to being in space. Like, as cool as we think it is to go to space. And I think it's incredibly cool. Our biology seems to hate space as much as we love it. There are a bunch of reasons, all of them boiling down to space being not Earth, the place we're from, with environmental conditions we're adapted to. I've talked in previous episodes of May Contain Science about how our bodies do not like space, but to recap a couple of the big reasons. A ton of different cell types in our body respond to mechanical force, and, well, those forces change when you're in the microgravity of space versus the lovely 1G of Earth. If your cells typically respond a certain way to a specific kinds of mechanical force, and those specific kinds of mechanical force aren't exerted on them because they are in microgravity, those cells may not give you their typical response. I mean, change the input, change the output, right? You can think of muscle tissue and how you build strength by lifting weights or doing resistance training. You know, exerting specific types of mechanical force versus the opposite. Like in space. The other big thing is that on Earth, we experience less ionizing cosmic radiation. Not none, but less than in low Earth orbit without protection from Earth's atmosphere. That radiation causes DNA damage, which is bad because it increases the risk of cancer. But DNA damage can also prompt cells to go hyperprotective with a program called senescence. The idea is that the cell goes, I can't become cancerous if I can't replicate in the first place. And then it turns off its ability to ever go into cell cycle and replicate again. That's very useful and clever because it does work to prevent that cell from becoming cancerous. But if you can't replicate because you're senescent, well, that's fewer cells that are capable of replication, and senescent cells, as it turns out, also behave differently from non-senescent cells in other ways. They tend to send out pro-inflammatory signals. They're a little aggro. A lot of these same mechanisms, senescence, cancer risk, are part of normal human aging as well, as we accumulate DNA damage over time and as our cells run out of replication lifespan and also become senescent from that. So that's why sometimes people talk about space causing accelerated aging, and that's from the cosmic radiation exposure and the other stresses as well, like microgravity and even social isolation on the space station. Things like exposure to cosmic radiation and exposure to microgravity affect every system in the human body. It's not like there's one organ where cells are taking DNA damage and just don't care, for instance. But different organs respond differently. Your bones aren't getting the mechanical force they're used to because you're in microgravity, and you start losing calcium from your bones. That hits your kidneys, which are in charge of maintaining your fluid, calcium, and salt balance, and you're in space, so your fluids are everywhere, and also now there's too much calcium in your blood, and the kidneys try to filter it, so they're just miserable. The fluid compromise in microgravity is an overall decrease in your blood volume, and your heart is supposed to pump that blood, and with the lowered orthostatic and arterial pressures, that's less work for your heart, so it gets weaker. along with all other sorts of musculature because they're not being exposed to specific mechanical forces. Meanwhile, the liver starts storing fat in ways that on Earth we usually see with metabolic dysfunction, and even the brain and eyes undergo changes. Astronauts report brain fog as a result of space travel, and nearly 2/3 of all astronauts end up with some degree of nearsightedness after extended stays aboard the ISS for reasons we're looking into but haven't fully figured out. We want to go to space because space is incredible, but our bodies hate space, and each of these organs becomes dysfunctional in its own way, with a lot of them feeding back on each other. We've studied these problems for decades in various models. Some we do in simulators on Earth, sometimes we send lab mice to space, so we have animal models, sometimes we get data from the astronauts, sometimes we do cell culture, but with specialized cell culture setup, because if you just had open petri dishes, you'd probably be okay for a little while because the surface tension of that little liquid culture medium there would keep it stuck in the dish. But the moment you had to, change the cell culture medium, it'd get real messy. So, we have specialized space cell culture, and that's where we come to our mini organs in space, our organoids. See, the short answer to our big question, why are we sending these organoids to space, is that we're sending mini organs to space because that's the way we do a lot of cell culture on Earth these days. Here's the classic cell culture that we would do in that petri dish we saw before. The cells line the bottom of the dish, adhering to some layer of collagen or other thing over the plastic, and you end up with one layer of cells, like cobblestones on a road. We call this a monolayer because one layer. You can do that with a single cell type, like just neurons, or you can throw, say, neurons and brain immune cells, microglia, together because cells communicate with their neighbors. They like being in the dish together. But in our bodies, cells aren't in a two-dimensional environment like that. They're surrounded by 3D scaffolding. We're 3D. And that scaffolding is called the extracellular matrix. And when you put this extracellular matrix, this scaffold, into a dish and let cells grow over and around it, you'll get a 3D culture. They clump together, usually into spheroids like you see in this diagram, but instead of being in cobblestones or like a carpet kind of thing on the bottom of the dish, like in 2D culture. Cells in 3D culture tend to be better at maintaining their cell-specific functions. Liver cells are better at metabolizing. Neurons are better at electrophysiological signaling, and so on. They tend to live longer in culture as well. So, cells in this clumpy 3D culture do better because it's more like the environment they're adapted to. That might feel familiar. And we get better science because the cells respond in ways that are a little more representative of their response in the human body. And it's likewise when you add the multiple cell types. It's more like the real body, so the cells are more representative. When we send a heart on a chip to space, it even has a little fluidic setup because heart cells are used to mechanical force from blood flow. Once again, it's more like the real body, and the data from that cell culture is more representative of what happens in the body as a result. So, we're sending mini organoids, organs, to space. We take human cells, program whatever those cells used to be to turn back into stem cells, which, by the way, turning cells back into stem cells is an awesome thing we can kind of just do. But that's for another day. So, then we give the induced stem cells specific recipes of signaling molecules on an established schedule because while the cell culture nerds were figuring out how to make cells happy, even though they're in a petri dish and not in your body, and teaching them how to clump together in their scaffold, the stem cell nerds have been at work figuring out how our bodies direct stem cells to transform into the specialized cells we have in our bodies. And oh my gosh, the stem cell nerds have figured out so many recipes for turning stem cells into liver cells, neurons, heart cells, kidney cells. Okay, so now we have our neurons or our heart cells or our kidney cells or our liver cells, and typically we have to do the stem cell to specialized cell process separately for each type of cell we'll want to include in our little mini organ. So, when these researchers, like the ones who just sent brain organoids to space and published in Stem Cell Translational Medicine, want to make organoids with both neurons and immune cells, they have to separately differentiate the neurons and the immune cells, then let them come together in culture before putting them on a rocket and yeeting them into space. And when we get all of it right, these cell culture systems allow for highly reproducible results that are more representative of what would actually be going on in humans. Because data from individual humans, literally the most representative. Whatever's going on in that human's body is, by definition, what goes on in a human body. But it's also not the most reproducible, controllable science. Like, the Kelly twins are great. But most astronauts aren't twins where we have, like, one on the ground and one we send to space. And they're not astronauts anymore. The Kelly twins retired. Like, Scott Kelly's writing a memoir. Let him live. We can't just do the most representative science on humans in that way. But then on the other hand, 2D cell culture, the traditional way we've done it, it's super reproducible. We've just talked about the ways in which it's not representative. Our cells don't live in a 2D environment all by themselves. So, here's our compromise. And now that we've sent these organoids to space, with more on the way, have we learned from them? Well, admittedly, we're still pretty early days. Most of these models, like the liver and the kidney stuff, are still being established. The liver's going up in early 2025 next year, proving that we can still successfully get these more complex cell cultures to work in space. It's not a trivial thing to do because these are complex setups. But we are getting some results. Recent studies with hearts on a chip seemed to much more closely approximate the cardiac dysfunction that we see in astronauts compared to some of those earlier cell culture studies. And now, as a result of those hearts on a chip, we have gene expression data from that approximate cardiac dysfunction, and that might give us some clues as to what's causing the problems that we see in our astronauts. Recent brain organoid studies yielded signs of accelerated maturation, although not necessarily accelerated aging, and we're hoping we might learn more from studies with another research group that's due to be published soon. So, as our spacefaring ambitions become farther reaching, we do have to take care of our spacefarers. I mean, all things considered, our bodies do surprisingly well in low Earth orbit, but we still don't fully understand our limits, let alone how to address them for the survival and well-being of our astronauts. And sending little mini organs to space may sound silly, but hey, if it helps those astronauts, safe travels, little brain. Enjoy space.